be back here in Houston. So as Shannon mentioned, I'm going to try to give kind of a big picture overview of how research has progressed over the past 17, 18 years. But in particular, spend a little bit of time talking about our newest research initiative, which is CF Labs, our own lab where we're doing research of our own and supporting and collaborating with researchers and companies all over the world to expedite and accelerate chordoma research. So I guess before getting into that, just a little bit of background, and I think today is really emblematic of this for me, is that chordoma really just affects a total cross-section of society, people of all ages, all backgrounds. I just think about what life would have been without chordoma and how many different types of people I never would have had a chance to meet. And so to me, that's a little bit of a silver lining, and today is very much emblematic of that. Is, all of us have such different backgrounds, and yet we're connected by this thing and a part of this club that none of us asked to be a part of. But nonetheless, there's a lot of good that can come from it, and both on a personal level, but I think also we're, as I'll talk about, on a more global level in terms of advancing research for this disease, and also maybe generating some insights that could be beneficial for other diseases as well. So my journey here and journey with chordoma started 18 years ago. As I just mentioned, I was an undergrad at Duke at the time, and was diagnosed with a skull-based tumor, which we didn't actually know what it was at the time until after having it resected, and it was only at that point that I learned that it was a chordoma. And I'm sure, like all of you, the statistics that I read were difficult to swallow in terms of seeing the high rate of recurrence and the average survival following diagnosis. And for me, this kind of lit a sense of, I basically lit a fire, and I became determined to try to do anything in my power, if there was anything in my power, to change those odds. So as I had shared with some of you at lunch, very serendipitously, it turns out that the only researcher at the time that had a grant to study chordoma, of all places, was at Duke, where I was studying as a student. And this was kind of presented this kind of incredible golden opportunity to try to do something. So to make a very long story short, I ended up joining his lab, becoming enthralled in the research. But it very quickly became clear that there was only so much that one lab could do, and that to make a real impact, we needed an effort that was much larger than this one lab, that would ultimately involve many different labs and companies bringing their expertise to bear on the disease. So at the same time, while I was working in the lab, I'd also connected with a number of other patients and family members through an online support group. And I don't know if Bill, you probably remember the MSN support group. So before Facebook and now Chordoma Connections, patients were connecting on an MSN group, and several of us had gotten together and were connected through this MSN group, and I think shared an interest in trying to do something in recognizing that something needed to be done to advance research and improve outcomes for those of us who are affected by this disease. So the first time I'd actually ever met another chordoma patient was in the fall of 2006 in Washington, D.C. at this conference of the National Organization for Rare Disorders. So I didn't realize this, there's like 10,000 different rare diseases, and for many of them, patients have kind of coalesced and formed organizations to try to provide support or advance research. And they host an annual meeting. So we decided to meet there at this conference. And first of all, just connecting with one another was, I think, very inspiring and comforting to meet other chordoma patients. But also it was really inspiring to see what had been done, what other patient groups had done, around small communities of patients, how by coming together, by joining forces, pooling resources, pooling efforts, they've really been able to move mountains for other equally rare diseases. So we left this meeting resolving to one another to do everything in our power to try to improve the odds and create a better future for those of us affected by this disease. That ultimately led to starting the Chordoma Foundation, which truly started off as a kitchen table operation. So, I mean, this was one of our early board meetings. So just families around a kitchen table. This was one of our first board chairs, Heather Lee, who some of you may know, some of you may have heard her story. Her young son Justin was diagnosed with chordoma when he was 5 and had about a 7-year journey with the disease before ultimately passing away. But Heather and her entire family and network were very instrumental in helping to get the foundation off the ground. Heather had a background in nonprofit management. None of the others of us really knew what we were doing, to be quite honest. We were putting one foot in front of the other. But it was very clear that there was an organization that was needed to raise funds, to coordinate research, to provide support, et cetera. And I'll talk a little bit more about some of what the foundation has done, but basically for the last 17 years, this has it's kind of morphed from a passion into a life's mission and a career, and I never would have expected that to be the case, but the foundation has grown and evolved in ways that so far surpass what any of us had thought was possible. You know, when this picture was taken, there has been so much progress in research and in terms of improvements in care. Obviously, there's been a tremendous coalescing of the patient community, and today is evidence of that. So I feel like there is so much good that has come from this journey. I had a really good run of 16 years tumor-free. And I think in retrospect, that's a big lesson, is that statistics only apply at a population level. Each individual's story is unique to them. So, I think there aren't, I guess as we all know, you're probably never out of the woods with chordoma, but the number of people who recur after 15 or 16 years is, it's small, even within, the population of chordoma patients. I was very fortunate a year and a half ago to have a successful surgery, and now my wife was 8 months pregnant when I had my surgery, but now we're very lucky to have a 1.5-year-old and a 5-year-old at home, and life has continued to go on and evolve in beautiful ways. And so I share this background simply to say that the statistics, yes, they tell us that you probably never get out of the woods with chordoma, but I think early on in my journey with this disease, I think I fixated on the statistics, and it was very difficult to allow myself to make plans beyond my next MRI 6 months or a year later. But I'm so glad that I have, and that I think maybe it was after 7 or 10 years that I started to kind of allow myself to fill in a picture of what a future could look like. And now I'm blessed with the family that you see here, which I feel like is something that I never would have imagined getting to experience when I was first diagnosed, but also now having had a recurrence and now having had, having a family, I feel like the stakes are that much higher. And so it motivates me. I'm not just fighting for myself anymore, but I'm fighting for them. And it is kind of engendered just an even greater sense of urgency that I think we all feel. And that is something that we bring to the work every single day at the foundation. So just quick overview, what does the foundation do? I think probably many of you are aware, but our mission is to improve the lives of those affected by chordoma and lead the search for a cure. And there's 3 pillars to this mission. So you are all, I think, well aware of our patient services, which you've heard about from Andrea and from Kimberly, and days like today are a part of that. Another important pillar is healthcare improvement, and by that we mean helping to educate physicians about best practices and even define what best practice is, because I think thankfully now there is more consensus about what optimal treatment is, but 10, 15 years ago that consensus wasn't there, and patients would, you'd go to one center and you'd be told one thing, you'd go to another center, you'd be told something totally different. generating, getting leading clinicians in a room, putting their heads together, hashing out, right, what do we know and what do we actually still not know? What do we have enough evidence to really get behind in terms of optimal care, and then where are our knowledge gaps? It's really important for improving outcomes with the technologies and therapies that we have today. So we've organized a number of consensus meetings and facilitated the publication of several consensus guidelines, which we hope are helping to standardize treatment for the disease. But really, the crux of what I'm here to talk about today is research. And while there definitely are just tremendous advances that have been made over the last couple of decades in treating the disease with surgery and with radiation, the reality is that for patients who recur and whose disease progresses past surgery and radiation, the options still are really lacking. There has been some progress made, which we'll talk about, but this is a summary of systemic therapy options. And what basically the take-home message here is that there are some therapies where small numbers of patients benefit or respond, but for the most part, effective systemic therapies are still lacking. And so once you've maxed out surgery and radiation, once the disease has spread, unfortunately, we're still living in a world in which options are not good. And there's a lot of research that needs to be done, but this is at the heart of our research strategy. Is what we're trying to do is facilitate and speed up the development of systemic therapies that can control recurrent disease, that can address tumors that have spread throughout the body and are no longer amenable to surgery and radiation. Eventually, though, perhaps a systemic therapy could be used in the adjuvant setting, maybe in lieu of radiation, maybe even before surgery and radiation, to shrink a tumor and maybe make surgery less risky. Maybe even we get to the point where systemic therapy takes the place of surgery or radiation. That's in all likelihood a long way off. But I think the other thing that we've recognized and that is kind of essential to the strategy that we've built is that it's unlikely that there is going to be a single solution to chordoma from a systemic therapy perspective. We know that cancer is heterogeneous, and that is the case for chordoma as well. So there's likely different subsets of chordoma that are going to require different systemic therapies. It's also the case that in many cases, successful systemic therapy paradigms involve combinations of multiple different drugs. So not a single drug that solves the problem, but a combination that can address multiple different vulnerabilities or prevent the emergence of resistance. And then we know that cancer evolves, and maybe a tumor responds for a period of time to a particular systemic therapy, but then another one is needed. So what this means is that there's no way that a priori, from the outset, we can predict that there's going to be one systemic therapy that we can put all our eggs in that basket. We have to create the conditions to make it possible for research to be done to lead to the discovery of multiple systemic therapies. And when you think about it from that perspective, that I think has a significant, that shapes how we kind of derive a strategy for attacking this disease. So where are these better systemic therapies going to come from? Basically, there's 3 different potential paths. So there's about 3,000 drugs that are already approved that are in the pharmacy right now. 250 of those are for cancer. Those could be repurposed for chordoma. There's about 7,000 drugs at any given time that are in development. So these are drugs that that various pharmaceutical companies across the world are working on developing. They're in clinical trials. About 1,300 of those are for cancer. And those, while a doctor can't prescribe them now, they could, a huge amount of money has already gone into developing those drugs. We don't need to reinvest that money, but we could potentially, if the right data is generated, start up a clinical trial with one of those drugs for chordoma patients. Or if the biology tells us that there is a particular target in chordoma that is not amenable to repurposing the existing drug or repositioning experimental therapy. That might tell us we have to develop a new drug. That's an entirely different process with different financial requirements, et cetera. We don't know at the outset which of these paths we're going to need to go down. We have to be led by the biology of the disease. But what we can know is that for each of these paths, they're going to have to traverse multiple different areas of research. So at the end of the day, there's going to have to be clinical studies that are going to be done to, in patients, determine which of these, whether the therapies are effective. In order to be able to design and justify those clinical studies, there's translational research that needs to be done. So largely this is work that's done in cells and mouse models of the disease. If the biology is telling us that there's new drug development that needs to happen, that's an entirely separate area of research that needs to be done. But all of it basically stems from an understanding of the biology of the disease and identifying therapeutic targets. A therapeutic target is basically something that's unique about the biology of chordoma that could be targeted with a therapy. So that's perhaps a vulnerability that the tumor cells have that normal cells don't have, for example. So something that the tumor cells depend on that healthy cells don't depend on. That could be a therapeutic target. Another example might be something that the tumor cells produce or express. So maybe a particular protein that's on the surface of the tumor cells that normal cells don't have. That could be a target. And there's a whole group of new therapeutic classes that target proteins on the cell surface. So maybe people have heard of CAR T cells or antibody drug conjugates, et cetera. There's really incredible technologies that target proteins on the cell surface. So we could find, for example, a protein on the chordoma cell surface that's unique to chordoma. That could give us a foothold for a targeted therapy. To do that target discovery research requires a set of basic resources, things like tumor tissue, cell lines, animal models, the kind of the basics of what a cancer research lab studies. And if those things don't exist, then there's no way to move on to the target discovery research. So to solve chordoma, ultimately to bring therapies to patients, there has to be progress across this entire continuum. Progress in every single one of those areas is necessary, but not on its own sufficient. So you can't just invest in one area and kind of hope for the best. You have to take a start to finish, end-to-end approach. And that's what we've done at the foundation for the last 17 years. We recognize basically is that progress in each of those areas requires several key ingredients. And the details are going to vary depending on the project, depending on the area. But in general, requires people. So people with the right expertise, deep subject matter expertise in developing disease models, for example, deep subject matter expertise in various approaches to understanding the biology of the disease, to drug discovery, to translational research, to clinical research. All of these are very, very specialized areas of expertise that don't reside within one individual, within one lab, oftentimes not within one institution. So that necessitates building a network of researchers that have all of these different complementary areas of expertise and making sure that they're linked together, that they're sharing information, that they're talking, that they're coordinating, et cetera. Sometimes though, what we have observed is that the researchers who have that cutting edge expertise, they're not necessarily intrinsically interested in chordoma. They're an expert in some signaling pathway or some technology to understand cancer. So there's this mismatch between the people who have interest in chordoma and then people who have deep subject matter expertise, and then the labs that have the capabilities to actually do the research. So it doesn't always reside in the same place, and so creating a critical mass. And I'm so glad that Dr. Raza is here because MD Anderson is one of the few places in the world where there is a critical mass of basic scientists, of translational researchers, of clinicians. We've tried to create that in a virtual sense and link up researchers who have those, the right expertise with others that have the appropriate capabilities. And then there's an additional ingredient, which is, okay, you can have expertise, you could have capabilities, but if you don't have cell lines, animal models, tumor tissue data to study, then you can't go anywhere. And so solving that problem was really a major focus of the early days of the foundation. And then, of course, all of this research requires funding for it to happen. So these are basically the ingredients that we as a foundation have tried to put in place. And we have been very fortunate to be met with a great deal of interest from researchers, researchers and clinicians, both those who maybe started off with an interest in chordoma, but as well as researchers from outside of the world of chordoma who had complementary expertise or capabilities that we've been able to bring into the field. So over the last 17 years now, there has been this very vibrant research community that has coalesced. Dr. Raza and other colleagues from MD Anderson have come to our research workshops. It's just incredibly motivating and inspiring to see the collegiality and the information exchange that occurs. And then, in between these research workshops, we're kind of working to kind of mediate communication and collaboration between these labs. Over the last 17 years, we've financially supported with research grants, projects in over 30 labs across the world. As importantly, if not more importantly, we've built repositories of tumor tissue and cell lines, et cetera. We've provided those resources to over 100 labs across the world. And so now we've kind of gone from this, really what was a desert and a very, very nascent field to now a field where some of the top research institutions in the world are very actively working on chordoma, and there's more and more progress that's being made. I think it's important to note that all of this is fueled by this community, by people doing walks, doing runs, doing letter writing campaigns, by contributing in a volunteer capacity. We are a small community, and what that means is that everyone can make an outsized impact. And it's, as I mentioned at the outset, just the progress that this community has been able to make by coming together so far exceeds what we would have imagined at the outset. So where have we come over the last 17 years? We've gone from having basically no research enabling resources, no tumor tissue, cell lines, or animal models, to now having abundant resources. We have 26 chordoma cell lines that have been validated and are publicly available to the research community. We've got a dozen mouse models of the disease. We have a biobank with 250 tumor samples. Almost nothing was known about the biology of the disease, and there were virtually no therapeutic targets that had been identified at that time. Now there's more than 40 potential therapeutic targets that have been proposed in the literature, and a lot of those point to drug repurposing opportunities. So maybe to drugs that are already in the pharmacy or drugs that are being developed that could be applied to chordoma. But the other really interesting thing that it points to is an Achilles heel that chordoma has, and probably many of you have heard about this gene brachyury, which is not only a diagnostic marker of chordoma, but the key driver of the disease and its greatest vulnerability. There are no drugs right now that target brachyury, but great reason to believe that if such drugs could be developed, that they could be very beneficial for chordoma patients. So at the outset, when this was discovered, brachyury was assumed to be an undruggable target. And I won't go into the technical details as to why that is, but very, very difficult, if not impossible, to develop drugs against. However, over the last decade or so, there have been a number of technologies that have emerged that start to make it conceivable to develop drugs against brachyury. And then the question has been, or our challenge has been, to be able to stimulate research, get researchers and companies interested in applying those technologies to brachyury. And just delighted that there has been interest. We've been able to raise some money and fund a number of projects, and now there are a number of projects ongoing, both in an academic setting as well as in companies. When the foundation started, there were no drugs that had ever been tested preclinically. And what that meant was that it was very difficult, if not impossible, to justify clinical trials. But now we've tested over 80 drugs in mouse models of chordoma. 10 of those have shown very strong efficacy and have now either have or are now moving into clinical trials. When the foundation started, there was one clinical trial that had ever been done. And now there have been 11 clinical trials that have been initiated thanks to the work that this community has supported. So here they are. Some of them are early stage trials testing the safety of drugs, and others are testing the efficacy of drugs. So some of them are ongoing, some of them, several of them have been completed, several of them are currently ongoing, and then several more are in planning. But I think the point here is that this is a growing pipeline, and several of these drugs are showing some benefit in patients. And I think in the coming years, as results from these trials read out and are published, I think we're going to start to see the toolbox of therapies begin to increase. Importantly, though, these drugs have been tested for the most part as single agents, and so part of what's coming next is being able to test these drugs in combinations, first in the lab and then ultimately in the clinic. So here you see this trial, pemetrexed and pembrolizumab. So these are two drugs that on their own have shown some degree of efficacy, and there's rationale to put them together. And so we've just committed to fund a clinical trial testing these two drugs in combination with the hope that that increases response rates. So I think all of this is great reason for optimism. I certainly feel much more hopeful now than I did 17 years ago. However, the reality is we need to go faster. Even though I think Chordoma, maybe some of us have been fortunate that the natural history of the disease, the tumor progresses relatively slowly. This is a race against time, and we are constantly looking and feeling a need to go faster. So essentially what we've recognized is that supporting research in academia and industry is tremendously important. There's incredible progress that can be made and that has been made. However, there's certain areas of research that are sometimes difficult to do in academia or industry, that they are either inefficient, they're insufficiently incentivized, they just don't align well with academic interests, or in the case of companies, there's no way that the company is ever going to make a profit off of the research. But yet that research still needs to get done. And so thankfully, over the last number of years, the ability to, for an organization like ours to get research done has really been democratized. There's contract research organizations that we can go to pay for research to get done. But also there's lab space, basically on-demand lab space that is designed for early stage biotech companies. And so a couple years ago, recognizing the need to be able, for example, to test drugs in cells very quickly without having to go through a process of finding a researcher who might be interested and awarding a grant, et cetera, just being able to answer a question quickly. We set up our own lab. It's based in Durham, North Carolina, in a facility called BioLabs, where basically we rent lab space. It's a fully equipped lab with all the equipment that we need, but the great thing is we didn't have to make the capital investment, the upfront investment to buy all the equipment to furnish the lab, to staff the lab with kind of the health and safety and facilities personnel. All we have to do is rent the bench and bring the scientists. And so we started this as an experiment a couple years ago, basically to create capabilities, again, to allow us to answer questions quickly, but also to serve as a central resource to researchers and companies to allow them to answer questions more rapidly and at less cost. Just to kind of visualize what that looks like. So if you think about it, if you're a researcher, you've not studied Chordoma before, to start up an experiment in your lab, there's a huge amount of startup time and cost that's involved. You have to import cell lines, you maybe have to hire personnel, you have to learn the nuances of how you grow these cells, set up the assays or the experiments. So there's a huge amount of time and effort that goes into that. But the beauty of having this all centralized in one lab is that all of that startup work has basically already been done. Those are sunk costs. And so now we can go to a researcher at an institution that's never studied Chordoma before, but maybe they have an idea, they have a particular area of expertise, and partner with them, benefit from their expertise and their hypotheses, and run the experiments that they would want to run, but in a fraction of the time and in a fraction of the cost. And then importantly, yes, publishing is very, very, it's important to get that knowledge out there. It's important, obviously, for academic careers, but oftentimes the publication timelines do not align with our sense of urgency. And so we want to create acknowledge the importance of publication and create the ability for investigators to publish while also having a mechanism for to build upon the findings that are generated as quickly as possible from a clinical standpoint. So not wait for the data to be published to share it with a clinical team and or a company that might just go ahead and start designing a clinical trial based upon those findings. So what we see is occupying a space within the research ecosystem that has historically been vacant, that has been a gap, kind of sitting between the world of academia and industry and basically bridging the chasm between the two. Work in the lab has really picked up. We've seen a lot more demand for the capabilities of the lab even than we had imagined. I think just to kind of play this out, there's constantly new things that are being learned about Chordoma, but also constantly things that are being learned outside of the world of Chordoma that could be applied to Chordoma. And now we have the ability to, when there's a paper published by a researcher that's never heard of Chordoma, we have the ability to go to them and ask, hey, does this, what you've discovered, is this technique that you've applied, that you've generated, could it be applied to Chordoma? Now we have the ability to very easily act on the ideas that kind of come out of those conversations. So there's a big pipeline of projects that are either ongoing or that are in the queue to get started. And what's exciting is that there's already, just in the span of 2 years, some really meaningful results that are coming out of it. There's AACR is the world's largest cancer conference, and in the last 2 years we've had 4 abstracts presented at AACR with work that's come out of the lab. 2 of these were in partnership with companies, and then 2 of them were actually internal projects that we initiated, and one of them was with an academic partner. So here's just an example of one of the projects. Basically, just a kind of a quick anecdote. There was a medical oncologist at Mass General who had an idea about potential therapeutic approach for Chordoma. He did not have a lab of his own, so he had no way to pursue this idea. He linked up with an expert in this particular aspect of biology, but that expert had never worked on Chordoma before. So we formed a 3-way collaboration between the medical oncologist, the basic scientist, and CF labs. In the span of a couple months, we were able to test their hypothesis, which was basically involved testing a couple drugs in Chordoma cell lines. In fact, it turned out to be exquisitely potent. So these drugs were exquisitely, the cells were exquisitely sensitive to these drugs. And then within the span of a few months after that, we were able to take those same 2 drugs into mouse models of Chordoma. And here you see, so basically the colored lines are the volume of the tumors that are treated with drug, and the black lines are the volume of tumors that are untreated. So you can see that these 2 drugs have substantially reduced the growth of these tumors in mice. So that was presented last year. Now we're working on combining these 2 drugs, and we see, so the mouse studies are ongoing, but the studies in cells have shown that indeed these 2 drugs are synergistic. And so that's hopefully heading towards publication and potentially clinical trials. I'll just go over this very quickly. So we funded a project at the Broad Institute of Harvard and MIT. So this was a grant that we provided over multiple years, basically systematically looking at all of Chordoma's vulnerabilities. So like, what are all the things that Chordoma depends upon for survival? And they identified about a dozen different genes that are selectively essential for Chordoma. So Chordoma depends on them, normal cells or other cancers don't depend on them. Brachyuria, as you might expect, is one of those, but there's others as well. The ones with red dots are ones for which there are already drugs available. And so based upon this finding, we were able to work with this academic group to quickly test a couple of those drugs in cells and in mouse models. And then actually based upon that data, there's been 2 clinical trials that have emerged. So this is, I think, a light year, a quantum leap improvement over where things were when the foundation started, even over where things were a few years ago. What does the future look like? How do we continue to optimize the pace of research? So, this is, the lab does not exist on its own. We are filling a gap. We are augmenting what exists within academia and industry. And what we've observed is that there are basic and translational researchers who have deep subject matter expertise, and there's Chordoma clinical researchers who ultimately need to translate that into clinical trials. And historically, there's been, the path between the two has been very fraught. It's been, it's been a dirt road, let's say. Places like MD Anderson, where kind of all those experts are under one roof, you can start to get a little bit more traffic going and make more progress. Where we see, I think, real, and by progress, what does that translate into? That translates into clinical trials. So there's several clinical trials that Dr. Raza and colleagues have gotten started for Chordoma. There's funding that's needed for that, and so we've been, thanks to the Chordoma community, we've been in a position to provide some funding to support the work at MD Anderson and obviously other places as well. But now with the lab, we can develop a much deeper, more tightly integrated partnership where there's data that's being generated by the basic scientists at a place like MD Anderson or others. That is leading to hypotheses that can be rapidly tested in the lab, goes back to the basic science researchers and kind of iterates in a tight cycle. And then that informs clinical research and what hopefully leads to more clinical trials. And then importantly, what's learned in those clinical trials can get fed back into work in the lab as well. And so I think this is kind of the optimal model that we see going forward, is forming deep, tightly integrated partnerships with a handful of centers, MD Anderson being one. There's another center in Europe that we're similarly seeking to forge a deep partnership as well. And I think if we can do that, it's really going to unlock a next level of speed and efficiency. So I think very exciting times in the field of Chordoma research, more and more reasons for optimism. The pace of progress is continuing to pick up, and I think if we see each other again in a couple years, there will be a lot more progress, I think, to report as well. Yeah.
Have they engaged AI in helping development of these drugs?
That is a great question. It's very timely. So the short answer is yes. The main area is in brachyury drug discovery. So I mentioned brachyury has historically been considered an undruggable target. So why is that? The protein, so brachyury basically exists in the cell as a protein. It doesn't have kind of obvious places for a drug to latch onto. And figuring out how to get a drug to basically stick to the protein has been an impossible task for human chemists. But where AI really could excel is basically trying every possible configuration of compounds to latch onto the brachyury protein. So doing that exercise is actually, from a computational perspective, I hate to say trivial, but it's a computational exercise. And there's dozens and dozens of companies and academic labs that have different AI approaches to do that. The bottleneck, however, is testing the compounds that they come up with in a biological system to determine whether indeed they're doing what they hope they will. And that's where the lab comes into play. So we haven't announced it yet, but we're just about to announce a prize specifically for this purpose, to basically say, hey, anyone who has a computational or AI-enabled drug discovery technique or approach, take a look at brachyury, apply your algorithm to this difficult target. If you succeed in finding a compound that binds potently to this target, then we'll provide a substantial prize. And we'll do the work of testing whether indeed you've hit the mark. Because these companies, they don't have the ability to stand up the capabilities to actually test those compounds. It's a year's worth of work and hundreds of thousands of dollars, which they're just never going to do. But if they can just hand over their compounds, we can give them the answer and then provide a financial incentive. The hope is that kickstarts a lot more interest.